Hi, welcome to the C1.2 respiration content for IB biology. This is the SL content here, and then we have the HL content here. SL content will be in green boxes, HL content in blue boxes as we go through, and a bit more HL, quite heavy on the HL for this one. Just ideas for how you can use this information to get yourself a grade 7. Don't just watch the video and think that's revision, it's too passive. Key terminology, SL terminology in green, HL terminology in blue. Okay, so we should already know that ATP is the energy currency of the body. We've come across that many times in the specification. And we know that two of the phosphates that are attached to ATP with high energy bonds, they're unstable, and once you break those bonds, the energy is released. So high energy bonds, break them, energy released, this is how we get our energy. This reaction is exergonic, which means that it's energy releasing. You can see here that we've got the phosphate being removed, and this is an exergonic reaction. If we're doing the opposite, then it's endergonic. The ATP can be used for active transport, movement of cells, anabolism, so many things in the body. Okay, uses of ATP. So glucose is super high energy compared to carbon dioxide and water in your respiration equation here. We should already know that oxidation is the loss of electrons or hydrogen, and reduction is the gain of electrons or hydrogen. So whenever an oxidation happens, a reduction must also happen. So we can see that electrons are removed from the glucose that goes into respiration and given to the oxygen. The glucose is fully oxidized into carbon dioxide. And the oxygen accepts all of the electrons and hydrogens in order to allow it to become water within that reaction. You can see that the energy is released. That's the energy in the form of ATP. Okay, so respiration is split up into a series of much smaller reactions. So you might have been told that respiration is breathing in and out, but that's not true. Respiration is actually a series of chemical reactions or metabolic reactions that happen within a cell in order to create energy. So the first part we can see is glycolysis, and this occurs in the cytoplasm. Now glycolysis occurs, produces pyruvate, and then the pyruvate is then going to go into anaerobic respiration if there's no oxygen available, or aerobic respiration if there is oxygen available. Now, because glycolysis happens in both aerobic and anaerobic respiration, it's referred to as the common pathway. It occurs in the cytoplasm. It uses 6-carbon glucose to produce 2 3-carbon pyruvates. So we're splitting those carbons from the glucose, and we're producing 2 3-carbon pyruvate molecules for every glucose molecule that enters glycolysis. Now in glycolysis, there's a net gain of 2 ATP molecules because 2 are used and 4 are made for every glucose molecule that goes in. Now if you're doing HL, we go into a bit more detail about this later on. If you're doing SL, this is all you need to know. In anaerobic respiration, this happens in the absence of oxygen, which we should know from previous study. Anaerobic ATP production is called fermentation. And we can see here that it's slightly different in yeast and plants. So on our flow diagram, the pyruvate goes into anaerobic respiration. In animals, we produce lactic acid. In plants, we produce ethanol and CO2. You can see here that pyruvate is converted into ethanal and then ethanol. We lose a carbon dioxide there. In yeast, in plants, pyruvate is converted into lactate. And we're going to use reduced NAD to produce this, and then NAD is also produced. That's anaerobic respiration. If we look at aerobic respiration, there's a series of reactions. So we have the link reaction, we have Krebs cycle, we have oxidative phosphorylation, and we have a huge amount of ATP being produced in aerobic respiration. So your body would prefer to respire aerobically because it produces more ATP that way. And then we have a little comparison. So we have oxygen required and no oxygen required for anaerobic respiration. We need the mitochondria for aerobic respiration because you can see here that happens in the mitochondria. We don't need mitochondria for anaerobic respiration to occur. Aerobic respiration takes place in the cytoplasm and mitochondria. Anaerobic only happens in the cytoplasm. 34 to 38 ATP molecules per glucose are formed, and only 2 ATP molecules per glucose are formed in anaerobic respiration. Nice place to pick up some easy marks there with a comparison. Okay, so we're going to have a look at the things that can affect the rate of cell respiration. So, number one, temperature. There's an optimum temperature for respiration. At very high and very low temperatures, the rate of respiration is going to decrease. Now, this is linked to homeostasis, so this is why your body needs to maintain a constant internal temperature. If we increase the rate of carbon dioxide, the concentration of carbon dioxide, this decreases the rate of respiration. Increase in oxygen concentration will increase the rate of respiration because obviously oxygen is used in respiration. The glucose concentration, if we decrease that, then it will decrease the rate of respiration and vice versa. If we increase it, then the rate will increase. And types of cells as well. So some cells have a higher rate of respiration just because they have higher energy needs. So some cells will produce more ATP and therefore carry out more respiration than other cells. Here we've got a respirometer. So this is a practical that you can do. Now you can see here that you have some seeds in this side. You have some potassium hydroxide, which will absorb any carbon dioxide within there. And then we have here a coloured liquid and a scale, so we can see how far that liquid's moved. So what happens is the seeds will respire, they will take in the oxygen, and it will suck up this coloured water, so you can calculate how much or how far that water has travelled. And if you have the diameter of the tube, you can also calculate how much of the water is moving in, and therefore the volume of oxygen that might be used in a specific time by the seeds. Okay, on to HL. So if you're SL, you can stop watching now. So we're going to start off with glycolysis, and we're going to look at glycolysis in a lot more detail. Now, here's our glucose, and it's a 6-carbon molecule. Now you can see here the key says that the darker grey is carbon and the lighter grey is phosphate. So we've got glucose. The first thing that happens in glycolysis is phosphorylation, which means that we're going to add phosphates onto something. And in order to do that, we're going to take the phosphates from the ATP. As we do so, we can take the phosphate, we'll produce ADP, take the phosphate, we're going to add it on to the ends of the glucose there. So we make fructose bisphosphate. Now fructose bisphosphate is quite an unstable 6-carbon molecule, so it will split really, really quickly. This is lysis. So we have the lysis stage, and it splits into 2 3-carbon triose phosphate molecules here. Now, once we have produced our triose phosphate molecules, you don't need to know this bit, says here, not required. We have our oxidation. Now, the triose phosphate molecule is oxidized, and we talked before about if an oxidation happens, then a reduction must also happen. So we're going to reduce this NAD molecule to something called reduced NAD, or NADH+. Now the reduced NAD, we're going to save that for later. We'll need it later on. You can also see that there are dephosphorylations that happen. So we're taking the phosphate off of those triose phosphates, and we can add it back on to produce 2 ATP molecules. So you can see that there's 2 phosphates on the triose phosphate molecule, the 3-carbon triose phosphate. We'll take those, we'll add them onto ADP, and we'll produce ATP. So for each triose phosphate molecule, we're producing 2 ATP molecules. So therefore, we're producing 4 altogether, but we use 2 at the top here. So we say that there's a net gain of 2 ATP molecules in glycolysis. So we've made 2 ATP molecules, there's a net gain of 2 ATP molecules, we've produced 2 reduced NAD molecules, and we've also produced this 3-carbon pyruvate molecules, 2 of those as well, for every glucose that goes in. So for 1 glucose molecule, we make 2 pyruvate molecules, 2 reduced NAD, and there's a net gain of 2 ATP molecules. Okay, So back to this graphic here. So we've got our glucose moving into glycolysis. We've produced our pyruvate so far, and we know how that happens. We're going to have a little look at what happens in anaerobic respiration. So this occurs in the cytoplasm. The pyruvate's been produced, and then the pyruvate enters fermentation in yeast to produce ethanol. So we can see here you've got glucose producing the pyruvate, and then fermentation is occurring to produce ethanol. Ethanol is a 2-carbon molecule because this pyruvate has been decarboxylated here and here. So therefore CO2 is leaving. So we end up with ethanol, a 2-carbon molecule. We're also producing NAD. So the reduced NAD is oxidized to NAD. Okay, aerobic respiration. So the next thing that happens after this, so our glucose has gone into glycolysis, it's produced pyruvate. Now the pyruvate has gone to aerobic respiration because there's some oxygen available. This is going to occur in the mitochondria. So the first thing that happens after we've produced that pyruvate is the pyruvate goes into something called the link reaction. Now you can see here your pyruvate, 3-carbon molecule. The first thing that happens is it's being decarboxylated, so we're losing carbon dioxide. We're also producing some more reduced NAD, so for every molecule of pyruvate we get 1 molecule of reduced NAD. And then we're going to add in something called coenzyme A. That coenzyme A will combine with the pyruvate that has been decarboxylated, and it will produce something called acetyl coenzyme A. That's a 2-carbon compound. After the link reaction, it will enter something called the Krebs cycle. Now in the Krebs cycle, we can see that that acetyl coenzyme A is entering, and what happens is that 2-carbon molecule is going to combine with this 4-carbon oxaloacetate here. Now that produces a 6-carbon molecule called citrate. We have 2 decarboxylations as we go along, so we lose 2 carbons, which means that that 6-carbon citrate turns into a 4-carbon intermediate. As we go through the Krebs cycle, you can see that we're going to produce 3 more molecules of reduced NAD. We're going to produce 1 ATP molecule, and we're also going to produce another molecule called reduced FAD. Now that's very similar to NAD. We're also going to save that reduced FAD for later. So we'll save our reduced NAD for later, we'll save our reduced FAD for later, and we've already made some energy, which is useful. Now remember, for every glucose molecule that enters, this Krebs cycle is going to happen twice, because we made 2 pyruvate molecules, so therefore we made 2 acetyl coenzyme A molecules. They both went into the Krebs cycle, so this happens twice for every glucose molecule. My advice would be copy the Krebs cycle out, label it all, copy it again, label it all, keep doing it until you know it. Okay, the final stage of cell respiration. So we've done aerobic respiration, we've done glycolysis, the link reaction, and Krebs cycle. Now remember, we made the reduced NAD and reduced FAD. We made those in glycolysis, link reaction, and in Krebs cycle. We're now going to use them. So we've been making them and we've been saving them for later. Now is the time. So the first thing that happens, in fact, I'll just explain. This here is your outer mitochondrial membrane. There's also an inner mitochondrial membrane. Remember that that inner mitochondrial membrane folds lots. It's highly folded. To increase the surface area, it folds into invaginations called cristae. In between here, we've got the intermembrane space. Now we want that intermembrane space to be quite narrow, and that'll become, we'll see why later. And then we've got the matrix of the mitochondria. I'm not sure why it says mitochondrial membrane there. This is the matrix of the mitochondria here. Okay. So we've got reduced NAD that we've made and saved for later. What's going to happen is that will be oxidized and it releases a hydrogen. And that hydrogen is then split into its electrons and its protons. Now the electrons will enter the electron transport chain. So these are proteins embedded within this inner membrane. The electrons enter that electron transport chain and they move through each electron carrier in the chain. As they move through, they lose a little bit of energy, and that energy can then be used to pump these protons from the matrix into the intermembrane space. So we're pumping them against their concentration gradient, so there's a high concentration here and there's a lower concentration here. The electrons continue to move through, pump some more protons into the intermembrane space, electron moves, continues to pump protons into the intermembrane space. Now this means that a really high concentration of protons builds up in the intermembrane space, and that means that the protons can move through this ATP synthase, which is another protein and enzyme embedded in the membrane. So this provides a channel for the protons to move through. So the protons move down their concentration gradient back into the matrix, and as they do that, they provide enough energy for ADP and a phosphate to be combined to produce ATP. Now, once the protons have moved through into the matrix, our electrons have moved through these big electron carriers, moves into the final electron carrier, and the electrons move out. What happens here is the electrons and the protons recombine to produce oxygen. You can see that down here. So when we have the electrons and the protons combine to produce hydrogen here. So you've got your hydrogen here, and that combines with oxygen in order to make water. So remember that water is one of the products of respiration. That's how we make water. We say this buzz phrase here, that oxygen is the terminal electron acceptor. So oxygen is taking the electrons from the electron transport chain, and it's combining it with these protons to produce hydrogen, and then we combine it with the oxygen to produce H2O. That's everything we need to know about oxidative phosphorylation. Oh, and then there's also, when the protons move through this ATP synthase, we call that chemiosmosis, that process, because they're moving down their concentration gradient. And the final part, we've got the use of lipids as respiratory substrates. So glucose isn't the only thing that can be used to produce ATP, and lipids actually yield 20% more ATP than carbohydrates. However, the glycolysis and anaerobic respiration can only occur if the substrate is a carbohydrate. So the glycolysis and anaerobic respiration pathway can't occur if the substrate is a lipid. Fatty acids are oxidized in the mitochondrial matrix, and the acetyl groups can combine with the coenzyme A and continue the cycle.